Welcome to the next session of Animal Kingdom or Animal Classification. Now just recollect what we did in the earlier session. We classified the kingdom Animalia into two subkingdoms, namely Parazoa and Metazoa. And under this we had seen presence of only one single phylum, that's Phylum Porifera. Now the first thing that we must understand, Parazoa is a kingdom, or Parazoa is a subkingdom that links Protozoa with Metazoa. See, protozoans are unicellular animal-like organisms. Unicellular means one cell, and animal-like cells here, that is without cell wall and plastids and all that. So that group of unicellular organisms and this group of multicellular organisms, this is in between the two. Now why do I say in between the two? Because in this, though it's a multicellular organism, there is no definite formation of groups of cells in form of tissues or organs or whatever. That means most primitive multicellular organism can identify these parazoans as. Under which there is a phylum Porifera. Pori means pores and fera means to bear. So these are the organisms which bear pores, or these are called as porous animals. Porous animals. Now we can call them as animals because they come under kingdom animal. Porous animals, perforated animals. Now here the unique feature is it's identified as cell aggregate body pattern. Cell aggregate body pattern. What do I mean by that? This is aggregation, collection, gathering of cells. But each cell almost works independently. There's no central control, there's no central coordination. Every cell functions independently. So it is just a collection of different types of cells. Hence it's called as cell aggregate body pattern. It is no doubt a single multicellular animal, but still each cell behaves almost independently with reference to the function that's assigned to it. Different types of cells are there. Different types of cells with different functions assigned to them. They stay together, they work together, but each cell behaves almost as an independent cell. Now how do they stay together? They are held together, tied together, bound together with some kind of a skeleton. What skeleton it is? It could be made up of spicules, or it could be made up of spongin fibers, a kind of protein, spongin fibers. Spicules are spine-like things. There are different types of spicules, we'll come to that. Further, these spicules could be made up of calcium salts when they would be identified as calcareous spicules. Or they could be made up of some silicon material, so they will be called as siliceous spicules. So primarily they could be identified as calcareous or siliceous depending upon the material from which they are made. Further, they could be identified on the basis of their structures. That is, if it's a single spine-like thing, it's called as a monoaxon spicule or monaxon spicule. One single axis passes through it. It could be a triaxon spicule, 3 axis. It could be a tetraxon spicule, pentaxon, hexaxon, and so on. There's another type. This is one more type. This is called as an amphidisc. The disc-like thing on either side, held by this rod-like thing in the center. That's called as an amphidisc spicule. Keep this in mind. We will have another mention related to it in the later part of the description. So monaxon spicule, diaxon, triaxon, tetraxon, pentaxon, hexaxon, these are different types of spicules, or there could be different spongin fibers which hold the cells together in form of one single multicellular animal. Now the unique feature of Porifera is presence of cells called as choanocytes, or they are also called as collared cells. That's because if say this is a cell, it has a collar-like structure and a long flagellum. So cell with a collar, hence the name collared cells or choanocytes. That's a unique feature. Then as I said, there are various types of cells. There are cells called as amoebocytes. There are cells called as epidermal cells. There are cells called as scleroblasts, and so on. Amoebocytes, as the name itself suggests, they are amoeboid in shape, phagocytic in function. Epidermal cells are more or less flattened, this kind of cells. Scleroblasts are the cells which actually have a significant role in formation of these spicules. They secrete the material, they produce the material, they release the material which is required for formation of these spicules. So they are responsible for formation of the spicules. Okay, so different types of cells held together by a particular kind of skeleton, that is what the animal basically is. As such, we don't name this as a diploblastic animal, that is presence of two germ layers, but still we find that there are two prominent layers. The outer layer more or less made up of epidermal cells, inner layer made up of collar cells, again held together by a skeleton. Another characteristic feature, which is the diagnostic character of Porifera, is presence of a canal system. See, we have identified this as a perforated animal, porous animal. These pores, what we are talking about, are minute pores called as ostia. Very small pores are there, microscopic pores are there. These ostia, there is another relatively larger opening called osculum. As you can understand, ostia is a plural, large number of these. Osculum may be single or may be more than one at times. And joined by these or connecting these two is a single central cavity, that's called as a spongiocele. Cele, as I said, is a cavity of a sponge, hence the name spongiocele. Now what happens here, these fine minute openings, ostia, they are the inlets. which lead to this single central cavity, that's spongiocele. Spongiocele then opens through osculum towards exterior. That flow, it's a flow of water. Remember, all the poriferans are aquatic animals. Most of them are marine, a few are freshwater. Very few are freshwater, mostly they are marine. So it's an aquatic animal. So through ostia, the water flows into the central cavity, that's spongiocele, and spongiocele, the water contained in spongiocele is eliminated through oscula towards exterior. And during this flow of water, nutrition, excretion, or gaseous exchange, that's a kind of respiration, would take place. So the system, the canal system would help in nutrition, excretion, gaseous exchange, that is respiration, that flow of water. The characteristic feature wherein the ostia would be taken into consideration, these ostia are intracellular pores. That means this is a cell, this is a pore in it, so here is the nucleus, this is all cytoplasm. The opening that you are seeing here is the ostium. So such minute microscopic pores lead to the spongiocele, which then opens to the exterior through relatively larger pores called oscula. Then there are amoebocytes, there are scleroblasts responsible for formation of the spicules and all that. Other features. Let's consider each life process. As I said, nutrition with the help of that single central cavity and the openings, that is canal system. Excretion also with the same, respiration also with the same. Mind well, the individual cell can have its exchange. It can take in oxygen, give out carbon dioxide as per the requirement. So respiration would be with the help of canal system, but individually related to the cell. These are majorly sedentary, stationary, non-motile, do not move. They have a solid substratum, solid holdfast, solid base to which these stay attached. Non-motile, you can say. That's a so locomotion, the feature doesn't come in picture. Reproduction. Here some unique features can again be mentioned. Reproduction can be by vegetative means, that is asexual you can call it, or can be by sexual means. Sexual means in the sense gametic cells will be produced which will unite together, form a zygote which can start growing further. Now if you remember, in Phylum Protozoa I had made a mention about encystment as a kind of reproductive process. And then I said that instead of calling it to be a reproductive process, it rather makes the animal dormant or resistant. Some similar thing can be seen even in Porifera, and that's called as formation of gemule or gemule. What exactly it is? When the conditions are absolutely unfavorable, there's a drastic change in the salinity of water, there's a change in the temperature, there's non-availability of food, or any such adverse condition if it arises, the animal goes to formation of a gemule. What's a gemule? Gemule would be those amphidisc spicules. This is one spicule, another spicule, third spicule. It will kind of form a complete sphere, ball-like thing. And inside these would be the amoebocytes. So this ball, what you see here, is called as a gamule that can tolerate all possible adverse conditions. It can stay dormant, it can stay non-active, and once again when the favorable conditions are there, this can start developing into a sponge, into a porifera. That is how gamute helps. Rather than reproduction, it makes the chances of survival better under extremely adverse conditions. That is what you see in porifera. Now, this phylum is divided into different classes on the basis of the type of skeleton it has got. 3 classes I would talk about. Class Calcarea, class Hexactinellida, and class Demospongia. Now, how do we distinguish? Calcarea, as the name suggests, calcareous spicules are there. Majorly calcareous, at times even spongian fibers could be seen. That's one prominent feature by which we identify this. Other characters. There would be simple type of sponges. Simple, why do I say? Because just go back and recollect what I said about canal system. The canal system, there are different types of canal systems. There could be an Ascan type, Sycan type, Leucan type, and so on. This is the simplest type. Ostia directly open into spongiocele, directly opens to osculum. Sycan type is slightly more complex. That means, if say this is the ostium, this canal is branched, and here it leads to the cavity. This is the cavity. This is the body wall, kind of peripheral covering. So this is slightly more complex than Ascan type. It's called a Sycan. Leucan type is still more complex. That is, from here starts the canal. It bifurcates. Here there could be certain vesicles. This also could be seen. This is called as a Leucan type of canal system. In Calcarea, Ascan, Sycan, Leucan, all types of canal systems are possible. Complex canal systems are possible. These are exclusively marine animals. Respiration, nutrition, excretion by the very same canal system. Reproduction is majorly by vegetative means or asexual means, but it can even show sexual reproduction at times. Non-motile, as I have already mentioned. Example could be Sycan. Here it is rested on some suitable substratum. These are all ostia. This is the outlet, around which you can see tiny spine-like extensions that forms an oscular fringe. This is the simplest type of sponge that you see as an example of Calcarea, marine sponges. Then we will talk about Hexactinellida. As you must be knowing, hexa means 6. That means the spicules are hexaxon spicules. This is a hexaxon spicules. And further, these are made up of silicon salts, so siliceous hexaxon spicules, you can say. Okay, that's a characteristic feature of Hexactinellida. Siliceous hexaxon spicules are there. Nutrition, respiration, excretion by the very same system. Reproduction is mostly by asexual means, but at times even shows sexual reproduction. The example that we can talk about, these are also marine sponges, exclusively marine sponges, basically sedentary, but at times they may cling to a motile support. The base which they are growing over could be motile, and then they would move along with the animal on which they are growing, or the body on which they are growing. Example could be Hyalonema, commonly called as glass rope sponge. Why so? The animal is something like this. These are all ostia, inlets. There could be an osculum here. Now, out of the 6 spines that are contained in every spicule, 1 spine is usually elongated. And the spines being made up of silicon material, these elongated spines of the spicules, they have a transparent glass-like appearance. Hence the name glass rope sponge. Glass rope sponge. That is a characteristic feature of Hexactinellida. Then Demospongia. A few freshwater sponges are known from Demospongia, but otherwise mostly these are also marine sponges. Now here the canal system could be Ascan or Sycan type. Leucon is not usually seen here. A larger number of ostia would be there. Oscula also could be more than 1 in number. Reproduction is by vegetative or sexual means. Example is Spongilla, wherein this could be some submerged twig. The animal grows over it. A large number of ostia and oscula also would be more than one. More than one oscula, large number of ostia, and this is what it grows over. Now, when I earlier mentioned that this can show locomotion with the help of the base, the support on which it's growing. So if it's going on a twig, that twig can float on the water, and that's how it can get carried. But otherwise, individually, the animal is non-motile. That is an example of Demospongia. Spongilla is